questions, the notes. Okay. So, to draw out the promoter of Bax in a cell without damage, that will look like this. Have a line, then Bax on the end. So here would be the starting site right here, so it'd be +1. And then the Tata box, which is -25, -35. So that's at -25 to -35 base pairs. So then would be the p53 binding site. This is at -1600, -1625 base pairs. So this would be the promoter for the cell with no damage. And I'll come back to that. So actually I can do this. Okay. So this part would be the transcription factor, transcript F. This part would be the initiation complex, which I will just write IC. And then this part right here would be SS, starting site. Okay, so for the promoter of Bax in a cell with damage, it would look pretty similar, but so Bax on the end. Then we still have that starting site at +1 right here. And then Tata box with the same location. -25 to -35. And the P53 binding site, 1600, 1625. So same thing, transcription factor, initiation complex, starting site. But because this cell does have damage, that means P53 would actually bind here. So we'll just go like that. P53 is binding to the site. Okay. So the difference between these two promoter drawings is obviously that one has the P53 binding to it and one does not. So in this top one, there's no damage, so there's no P53 being released. So there's nothing binding to the P53 binding site, which means that Bax would not be actively transcribed. So because that's not happening, X goes to the starting site. So for this promoter, because it does have damage, the P53 is being released and is binding to the binding site and is causing the Bax to be actively transcribed and is causing it to do its thing, which is the function of Bax is to slowly begin the process of apoptosis, also known as programmed cell death, also known as cell suicide. So yeah, that's pretty much the difference between these two, is one has the P53 binding to it, one does not. One has no Bax being transcribed and one does. So what will happen to the cells in a little checklist, hold on. So what will happen to the cells in 1 and 2? So this cell, because there's no damage, there's no P53 being released, nothing binding to the binding site, no Bax being actively transcribed. So really nothing would happen to this cell. It would kind of go along with its normal everyday life and do its thing. So for this cell though, the one with damage, because something's wrong with it, P53 is released, binding, Bax is being transcribed, this cell would start undergoing apoptosis slowly, and it undergoes apoptosis, and it tries to keep any of the damaged cells from surviving. So what its main function is it sounds bad, killing itself basically, but it is meant to actually help. It's getting rid of every cell that has some sort of damage to it, or it could be cancerous or something. So this cell, nothing would happen to it. This cell would probably end up dying. Yeah.